So I'm José Vico. I'm going to share this session, this afternoon session. And so the first speaker is Shilpa Khatri from the University of California, or Merced University of California, and she's going to tell us about coral reefs, I guess. And I'm very excited to discover about corals.
Thank you. And I'd like to start by just thanking the organizers. It's been a pleasure to be here so far, and I'm looking forward to seeing the rest of the talks. So I'm going to be talking about pulsating soft corals. My name is Shilpa Khatri, and I'm from the University of California, Merced. So I'm going to do a little promotion. So this is our department. A lot of you have asked me about it, but it's a pretty new university, and we're always hiring. So we have faculty positions this year, as well as postdoc positions, so if you know anyone, or if you yourself would like to apply, you're welcome to ask me about it. Okay, so I'm going to be talking about soft corals. This is what they look like. And why are we even interested in these organisms? So these are animals that live in coral reefs. And most of the coral reefs and what you see in the news about coral reefs, they're talking about the stony hard corals. These are elastic bodies and they're soft, and they also live along those stony hard corals. And when people talk about acidification, they're talking about the death of the hard corals. And so what's been observed, and it's really unclear right now, is that the soft corals are starting to proliferate a little bit as the hard corals are dying off. And so understanding why they pulse and how they succeed is of importance to the coral reefs. Very few animals, so this is a specific species that pulses like this, very few animals pulse or have an active motion that's not for locomotion. So I will show one other species that we compared to at the end of this talk. But these are one animal that has this energetically expensive motion, and the question is why? And so biologists have been studying this, and they've hypothesized some things. So the most obvious might be that it brings in food for the animal. And so most of these animals are found in the field without food in their gastric cavities. So it's believed that they don't actually eat that much. They might eat a little bit. They do have a stomach, but it's unclear if they're eating at all or how much they're eating. More, it's believed that they get most of their energy from symbiotic algae that live on them. And so they believe the pulsing motion is helping to increase the photosynthesis of the algae that live on them, which provide them with this energy or carbon. And then finally, these animals the individuals live in these large colonies of hundreds of polyps. And so there's belief that maybe it's not just helping the individual, but the entire colony in some way. So these are the kinds of things that have been hypothesized. Our collaborators work in the field off the coast of Eilat, Israel, in the Red Sea. And so that's a map. They have an underwater PIV system, so they can look at the flows created by these corals. This is very different from the stuff I'm going to be talking about today in the sense that they can't look at individual polyps. They're able to look over an entire colony. So in this picture here, this is the colony of coral, and this is their laser and their camera system set up. These animals live about 5 to 10 meters deep, so these systems are set up by diving and actually carrying down the equipment. And this is what a colony looks like in the field. And the first thing I would like you to observe is how strong the background flow is. So there is a very strong background flow at the depth that these animals live at. And so it's really kind of an interesting question of what is the flow created by the individual polyps that's different from kind of the turbulence that's naturally present. And why are they spending so much energy on top of like just benefiting from their environment? Okay. So this is some of the data that was presented when I got interested in this area of research. So they had done some work in the lab, so these are experiments done in the lab, where they were trying to see if this could be possible, that it's helping the algae photosynthesize better. And what they did was some experiments where they had kind of natural conditions in the lab, as well as putting in conditions in the lab where they increased the oxygen in the tank to some very high level of oxygen. And oxygen is the waste from photosynthesis, and they believe that it's the limiting factor in the algae photosynthesizing. And so when they did these experiments, they were believing the pulsing motion was removing the oxygen and allowing the algae to photosynthesize better. And this is what they actually saw. So when you look at these two bars here, you're looking at corals that are at rest and then corals that are pulsating. And in black, you have the condition of natural conditions, and on the right in white, you have the conditions of high O2. And what you observe is when they're pulsing, they're able to remove more oxygen in the case of the ambient, the natural conditions, and have more photosynthesis. Furthermore, they looked at what happens as you increase light intensity. The dashed line here is non-pulsing corals, and the black line is pulsing corals, and you see they photosynthesize much more when you have pulsing behavior. And then this is what the kind of data they can get from the PIV in the field looks like. And you can see that there's a pretty big difference in the velocity fields. This is a measure of turbulence intensity. So you can see that the pulsing motion is creating quite a bit of turbulence that you don't get at, when they're non-pulsing, even though in both of these cases there was a pretty clear, strong background flow. So our goal, okay, so this is the one last piece of data I want to show from some prior work from that paper. And what's interesting is I've talked a lot about this photosynthesis stuff, but when they went down and observed at how long and when do these animals pulse, it's observed they pulse all day and all night, very clearly with a 1 or 2 hour break at sunset. And so there's a really open question to why are they pulsing at night if it's for photosynthesis purposes. And so it's not completely clear. So there's a lot that's going on with these animals. Okay, so what are we doing? We're trying to be able to look at the flows created by the individual polyp, kind of get a better sense of what's really going on in terms of the fluid flow, and then hoping to couple this down the road in the long term to actually looking at concentrations of O2 and CO2. So we're doing this by doing PIV in the lab, where we can actually get close in and validating 3D simulations. And so that's what I'm going to be talking about today. So here's an individual animal, so that they are called a polyp. They have 8 tentacles, so these kind of the things that actually move and create the pulsing motion, and then the base is called a stem. This is what our simulations are going to look like. I haven't plotted the stem here. We do have a stem, so it's closed. We don't include kind of the stalk of the stem because it's not really moving or creating flow. To give you a size, it's about ½ a centimeter from the center to the tip of the polyp, and it's about at Reynolds number 10. Okay. So we're doing fluid structure interaction where the coral has a prescribed motion, and I'm going to talk a bit about where we get this prescribed motion. We're actually using data and measuring kinematics from the animals from data we have from the lab. We're solving the Navier-Stokes equations, your conservation of momentum, your conservation of mass. You have a force from the coral on the fluid. That comes in that term. And we're using the immersed boundary method, which was developed by Charlie Peskin in the 1970s. Our Reynolds number, so I said the Reynolds number is about 10 for these animals in the environment. It's based on the length scale of the coral and then the frequency at which it pulses, as well as the density and viscosity of saltwater or seawater. So I thought I'd give a quick summary of the immersed boundary method. I'm going to do it in 2D, even though our simulations are in 3D, because it's much easier to explain. And so this is supposed to be a 2D tentacle. So this coral has 2 tentacles. You have an Eulerian mesh, a background grid on which the fluid equations are solved. You have a Lagrangian mesh, which represents the coral. So you have a set of points that's representing the motion of the coral. And then you have a force from the coral motion to the bulk fluid that's spread by integrating over the boundary and using the delta function. So that's kind of the basics. The force you apply is an elastic force density. So the numerical method, you advance the coral location based on this kinematic data. You compute the force from the corals onto the fluid grid. You do this by regularizing the delta function so that you can go from the Lagrangian grid to the Eulerian mesh. You solve for the fluid flow using the forces from the corals. We use a projection method, kind of a standard numerical method for this. And the simulations are done using IBAMR, which is a library written by Boyce Griffith. It's a C++ library that we kind of program in the motion of our corals and are able to use the library to solve all of this. Okay, so what does this look like? So this is, so to get our kinematic data, we have these videos, and we have a graduate student who went through and tracked all of a number of these videos by hand. So we track 6 points along the coral tentacle. We fit a polynomial at every single frame. We compute the arc length at those 6 points. So now we have the x position, the y position, and the arc length at all 6 points. We non-dimensionalize, which is going to allow us to average over many pulses and over many polyps. And then we fit polynomials for x and y as a function of arc length at each frame. And so when you do this for this one polyp shown here for 5 pulses, this is what this looks like. And you can, the red line is the polynomial fitting that's been done. And it shows you it's a pretty good accurate representation. We're looking at a tentacle where we're taking a cross-section kind of in the 2D plane. So that's how we're getting the motion. We make the assumption that it's not rotating in the 3rd dimension. And what we've observed is that the animal tends to stay in the 2D plane unless it's unhealthy. So these are animals that are quite hard to keep alive in the lab, and which is why we work really closely with our field collaborators. When we do stuff in the lab, we just take the animal out of the field, put it in the lab, do experiments for a couple hours, and then put it back in the field. And when they start to get unsettled, so when we've done a lot of PIV on them, when there's particles floating around, they tend to start to twist. And that's kind of a sign that they're not happy with us. So most of the time, they tend to pulse just in that one plane. OK. So once you have all this data, so I have 4 coefficients for 4 polynomials, or 2 polynomials, 4 coefficients. You have, at every frame, you have a number, a value for those coefficients. And so that's one of those coefficients here. And I'm going to just zoom in on that guy. And so this is what this looks like. And what we do then is we divide the pulse into a resting phase, a contraction phase, and an expansion phase. And so this is divided into 3 phases for each pulse. And here we have 5 pulses for this one polyp. And then we fit to each phase a 4th order polynomial in time to the coefficient. And so once we've done all of this, we can go back and reconstruct our motion. And that's what it looks like. So this is for one pulse. You can see the resting phase, and then the blue points are the data from those frames. Here you have your contraction, so you're coming in. And what you should observe is the expansion as a different behavior than the contraction. So you're coming in kind of like this, and then you kind of sink down and open in a different motion. Okay. And then we average over the 5 pulses by averaging all the coefficients of the polynomials. And you see that we have a very similar behavior as what we saw in that one phase. Okay. So that was for one polyp. We do this for a bunch of polyps, and then we average over all of it, and this is what it looks like. It doesn't look terribly different. The one thing I do want to point out is the resting phase looks slightly different, so I'll go back. Looks like I should have. So in this case down here, you're really splayed out, and you're almost, I was like concave down, while here you're not. And this has to do with different polyps tend to extend a different amount when they expand. And so there is some variation that's like very visually obvious there. The resting phase is about 34%, the contraction phase is about 29%, and the expansion phase is about 37% of the pulse. So you tend to contract a bit faster than you expand, and then you sit around for a good chunk of time in between pulses. And so these were average measurements taken over all the animals, just to give you some idea of scale. So the width of the tentacle is about 1/10 of a centimeter. As I said earlier, it's about 1/2 a centimeter across. A pulse is about 2 seconds. And the Reynolds number for these average guys is 10.66. And this is what the motion looks like when we put it into the immersed boundary method. So this is what we're going to be coupling with the fluid flow. Okay. So I wanted to talk a little bit about kind of the numerical setup before I showed you guys all the results. And so here, what I have are preliminary 2D simulations with kinematics that were based on stuff we had kind of from preliminary data when we were just trying out the first ideas. And so it's not the average kinematics I just showed. But what I have going on here is I wanted to point out boundary conditions and kind of how the solver works a little bit. And so what I'm plotting is vorticity and velocity vectors. And you can see that you are getting an upward flow where you have vortices shedding in 2D. You have Dirichlet boundary conditions on the top and bottom walls, and we'll have periodic boundary conditions on the side walls. And we set the domain large enough that they should not be interfering with the flow that's being created. So we can see quite a large domain. And this is what the adaptive mesh refinement looks like in 2D. It's much easier to visualize in 2D. So we have This is just showing kind of the levels of refinement, and then this is actually showing your grids. So you have finest refinement at points where you have boundary, like close to the boundaries, or where the vorticity is greater than 0.5, so where the magnitude of the vorticity is large, is kind of where we refine. We have 4 levels of refinement, and the ratio between each level is 4. And so this is a pretty intricate solver in that sense. And then here's a convergence study. I was like, your course to your fine, and we see that it converges as expected. So we see that we have second order convergence in velocity and first order convergence in pressure, and this is what we would expect. Okay, so yeah, I was like, I'm going to show you guys a 3D simulation now. So this is our simulation using our average kinematics, and what I'm plotting here is the vorticity contours and the velocity field vectors at the same time. And we have, in all the previous stuff, the stem wasn't included, but you can see now it's closed in. You do have this base that's creating flow. And so what's really nice about these simulations, kind of compared to, I'm going to show you some PIV data in the next slide, is that we can really see what's going on close to the tentacles and the behavior that's happening. And what you observe here, and I was like, it's not showing up super well at the top, is that you're pulling in radial flow from the sides, and then you're pushing the fluid upward. And you're doing this in a continuous motion. Even when I'm expanding, you tend to get an upward flow up here. So even though I'm doing this back and forth motion, it's slightly different, and it's different enough that it's creating a consistent upward flow. And I'm at a Reynolds number that's high enough to do that. And then there is, I guess, close to the top of the polyp here, you will see a little bit of backflow, so it's minimal, and that allows some mixing to occur interior to the polyp, which is important because it is trying to sample the fluid. This is experimental data from the lab. It's an individual polyp. This is actually really rare. We don't usually see individual polyps, and when we work with colonies, we spent a long time trying to get one of the animals to pulse and the rest not to, and they're very sensitive, and they don't like to do that. So we kind of were lucky to get this set of data. And so what we have here is one polyp that's pulsing. We have the laser sheet is centered at the center of the polyp, and so you're getting 2D velocity fields through the center of the plane. And you can really see this upward jet that we showed in the simulations here. And so qualitatively, we can see that we're getting pretty decent results. I'm going to show some quantitative comparisons in a second. And you do observe even more closely that you're pulling in fluid from the sides and it's pushing it upward, and you see the separation that occurs. So you have, it's like when it expands here, you see that there's upward flow up here, and then there's like circulation that's mixing interior there. And I just wanted to show you some PIV from a colony to show you that this is not unique behavior when you have an individual polyp. And so this set of data is set up so that your laser is centered at the center of the polyp. Like, okay, on the left here. And this data is not as well resolved as the previous data. It wasn't as well seeded as the previous data, and so it's averaged over 4 frames, while the previous data was done at every frame. And so, but you're still seeing that you get a very strong upward flow in the direction of the stem. So you're here, and as it expands, you have that separation. Yes?
What type of PIV is this?
It's 2D PIV.
No, I mean, is it pulse laser or is it time resolved?
It's a pulse laser. Okay, so, and I was like, and these plots don't show up at all. So you're supposed to be seeing streamlines as well as vorticity. And so I will just tell you what's going on. So these are from the simulations, and then I was going to show comparisons to the PIV below. And so what we see, what you should be seeing here, and you can kind of see, is that when you're about to start contracting, you have some stopping vortices right here. And then when you start to contract, and as you get close, so in this frame right here, the polyp is completely contracted, you formed vortices in the opposite direction of the stopping vortices from your rest phase, and you're now pushing those out towards the tips. And this is a vortex ring in 3D. And this vortex ring expands, and it draws fluid kind of up the center of it. So there's streamlines that are going vertical up the center right there. And then as you relax, you see that you get the vortex ring from before is still pushing fluid upward, but you're now forming those stopped vortices in the opposite direction, and it's circulating fluid close to the top of the polyp. Okay. And in the PIV, we see very similar things. The data is not as clean because it is experimental data. And so I was like, so you would see your stopped vortices in the first frame and kind of see the formation. And you see it here as the opposite vortices right there. And then it's moving upward. And it's not super symmetric in this case because the polyp is not perfectly centered. But. Okay. And then we wanted to actually quantify something to be able to look at plots and say, are we getting the right magnitude of velocities? Are we getting the same behavior as we move away from the polyp? And this is what we're trying to show here, that our model is doing, I would say, a fairly good job. And there's some differences that I'm going to talk about that we're going to try to fix. So on the right here, we have our 3D simulation, and what we're looking at is velocities averaged on 2D plates as you move some distance away from the polyp. And so we're looking at vertical velocities on the horizontal plates and horizontal velocities on the vertical plates. And these plates are about half a tentacle length. So the first plate is right above the polyp when it's contracted, and then this is half a tentacle length and a full tentacle length. And the same is true for this guy. This guy's right at the left when the polyp is fully extended, and then you have half a tentacle length and a full tentacle length. And these are your vertical velocities, and this is for the PID, and then this is for the simulation. And the maximum velocity for the average vertical velocity is approximately the same, so we do capture that quite well. What we don't capture quite well is as you move away, you have more decay of the velocity in the simulation. And the horizontal velocities, we have the same behavior. We don't match perfectly. We're not too far off. We're off by, I think this one's about 3 times as large as this one. And we believe this is actually due to the fact that we're using different kinematics in the two in the simulation from the experiment. So we're using the average kinematics from 5 polyps, 5 pulses on the right-hand side, while this is just one of those that was included in the average. And the Reynolds number is quite different between the two. And so the Reynolds number over, for the PID data is about 20, while the Reynolds number for our simulated polyp is about 10. And so we would like to redo these plots with the correct Reynolds numbers, and we think this will take care of the discrepancies. But overall, we're pretty pleased. Like, the data looks pretty good. And it allows us to observe what's going on. The model allows us to do a lot more than we can do with the PID. And so this is what I'm going to show here, is that we've done this 3D simulation, and with the PID data, we aren't able to observe what's going on within the polyp or close to the polyp. And while we can do that with the simulations. And so what we're plotting here is the picture, the movie I showed you at 4 different frames. And so as you're contracting, fully contracted, and then as you're expanding and at rest, and looking at what the velocity field looks like on the plate above the coral polyp and on the left-hand side. And what you observe is, so red here means a strong upward flow, and blue means a downward flow. And so you see as the polyp contracts, you have this very strong upward flow, and then as it expands, you continue to have, and I was like, it's not as clear here, so it's supposed to be pink, kind of outside that white ring, that you have a pretty strong upward flow kind of on the exterior near the tentacles, while you have downward and horizontal flow within the polyp. And that continues into the rest phase. And what you see from the horizontal flows, or you should see from the horizontal flows, is that you have a strong radial flow coming in. That's the red right there. And then as you expand, you get some backflow, but not a ton. And you have kind of a continuous flow coming in because you're continuously pushing fluid upward. And so it's significant biologically because it means you're always resampling fluid. So you're always bringing in new fluid. You're always having an exchange of fluid, but you're having some time where it sits kind of above the polyp to mix it up and resample it and take things out or bring things in. So if you're taking out oxygen or bringing in CO2. And so this is why this is important biologically. And of course, with simulations, we can change parameters that you can't change in the lab. And so the first thing we did was modify Reynolds number to look at some different things going on. And so the middle movie here is the actual animal regime, while on the left you have small Reynolds numbers, and on the right you have high Reynolds numbers. And what you observe is that all three of them, as the animal contracts, has this upward flow. It forms those vortices. But on the left-hand side, where you're at small Reynolds number, when it expands and reverses, the flow completely reverses, as we would expect. While for Reynolds number 80, you get such a strong upward flow that when you compare, I was like, the tentacles, the flow between the tentacles in, some buttons, okay. When you compare the flow between the tentacles, as it expands, you actually have more flow between the tentacles at Reynolds number 10 than you do at 80. And so it's suggestive that the animal has chosen to live a motion at the regime it lives in that's ideal for the things it needs to do. So it needs to resample the fluid. And we can see this a little bit better. So we plotted the average vertical velocity and average horizontal velocity. These are slightly different plots than before because instead of looking at those three plates, we're actually averaging in the entire volume of those three plates. And I think we want to go back and look at what's going on in these three plates independently, but we just started by looking at these. And what you see is that Reynolds number 10, and it's not, I was like, not super clear here, but for the Reynolds number 10 and up is kind of, 10 is where this bifurcation occurs, where you no longer have an average negative flow. So when you look at the average of vertical velocity, when you're above Reynolds number 10 and greater, you have a flow that's moving upward. While for Reynolds number below that, you do get a negative average flow. And you see kind of a similar behavior in the horizontal that corresponds to that, because if you're moving flow upward, you should be bringing in fluid radially. And so that's what you see going on here. And not only that, but when you, so the maximums of both of these occur when you're contracting. And what you observe in the horizontal is that it not just maximizes when it's contracting, but it kind of stays constant in the radial direction, averages, when you're above Reynolds number 10. And so you're continuously bringing in fluid in the radial direction at a constant speed, while in the case of the lower Reynolds number, you're bringing in fluid and then pushing it back out. Okay. And then this is some stuff we've just started doing this week. So we are adding in background flow, and I'm just showing some movies off the press, but there's a lot of stuff going on here, and we want to do some further analysis. So I'm not going to present any plots. But what we're looking at is what happens now if you add a strong background flow. How do things change? What's the modification? And how we did this was we have our box, our domain here, and we just set a shear flow on the top wall. At some speed, we started at 0. We ramp it up the first pulse to 2 centimeters per second for the movie on the right here, and we look at what happens. And what you see in this movie is what we would expect, that you get an asymmetry in the way the vortex, what you're supposed to see in this movie here, is that so when this vortex moves upward, it's moving to the right, as one would expect, because of the flow to the right. What we're trying to understand, and it seems like a very complicated behavior, is what happens to the maximum velocities and what happens to this upward jet. Can we look at these averages of this flow, and how is it moving fluid away from the coral, and does it still allow for mixing when you start adding in background flows? And we would like to look at different background flows. And it seems like depending on the speed of the background flow, there's a lot of variation. So it's not a monotonic behavior. It's just some complicated dynamics. And so this is what we're starting to look at. Okay. And I said I would tie this back to another species that also has an active motion that doesn't locomote. And this is the Cassiopeia, so this is an upside-down jellyfish. So these are animals that are able to move if they want to move. But usually, they find a place where the flow is really quiescent, and they settle down at the bottom of the ocean, and they hang out until they get unhappy, or there's more flow, and they're no longer happy with where they're located, and then they can move away. And so when they are settled at the bottom and are just sitting there, they also pulse, and they create an upward flow similar to the corals that we're looking at. What's really interesting is they're completely they live in a completely different regime and they have a completely different behavior. So their Reynolds numbers range from hundreds to thousands instead of the tens, like the ones we're looking at, and they live in quiescent flows instead of places with strong background flows. And the way they pulse is this bell down here is what pulses. It's a very thin, long, kind of flatter, round pulsing motion, and the bell pulses, and so it's axisymmetric. And so it's this part right here. And then it must pulse fluid through these oral arms. So these oral arms don't move. And it has to, so the bell must push the fluid through those oral arms, and so it's kind of like it has to push fluid through a porous layer, is how you could think about it, while the corals don't have to do any of this. And I'm just going to end by showing you some plots that argue that this does have this upward flow. So this is PIV data of the bell contracting down here, and you see that it's creating a strong upward flow through the oral arms, and the same as it expands back out. So this is what this is showing. And so it's really interesting to compare the jellyfish to the corals, because there's two of the species that do have an active motion that don't locomote, and they do create this upward flow, but they live in very different regimes. And so they're both successful at doing this with different mechanisms. Okay, and then I was just going to show the next step is obviously to add solutes to look at if the coral is creating or destroying concentrations, and this is what my graduate student is working on, developing numerical methods for this. But this was a preliminary study just looking at if we stuck a box of a concentration, what would happen, and what does the mixing look like. So with advection and diffusion, and you can see that you are like pushing fluid upward as one would expect, and you're pushing the concentration away. Okay, so today what I've shown is we've developed computational tools which allows us to explore these flows. We've done a lot of analyzing of these flows. What we would like to do is some more accurate modeling of solutes and maybe some particles that carry inertia. We would like to play a little bit more with these parameters that I've shown, so the background flows especially, and we would obviously like to study what happens with many polyps instead of individuals. And I was like, there should be one more slide. I will. Did that hide? Nope. I don't know what happened to it. It's supposed to be a thank you slide to my collaborators, so I'll just name them. So I work with field collaborators in Israel, so Uri Shavitz and Roy Holzman, and then Laura Miller and her graduate student Julia Sampson and undergrad student Dylan Ray have been working on the PIV data extensively and helping to analyze and compare with the simulations. And my graduate student Matea Alvarado and I have been working on the simulations. And then thanks to NSF for funding. Thank you.
One question?
Yes.
Oh, there is one in the back.
Maybe it's a little unfair because this is the work you're still planning, but
Yes.
aren't these things so close together that they are obviously interacting, the flows are obviously interacting, and more sort of pointedly, I've noticed in some of your films that there seems to be an anti-synchronization, many systems that are periodic in nature as a synchronized or anti-synchronized.
Right.
Aren't you missing a big part of the story?
Yes, so we're working on this. So Laura's graduate student has been looking at the synchrony, and what we can say is that it's pretty random. It's not anti or fully synchronized. So, she's looking for patterns in the data right now to understand this a little bit better. Yes, the flows are interacting. The point of my colony PIV was to show that even though we are looking at individual polyps right now, that even in colonies they seem to have similar flows, and it still has the same theme of this upward jet. And that seems to be true. We do know that their nervous system is probably somehow connected as well in the colony, or they're somehow sensing the flows, because we know if we touch one of them, it stops pulsing, and then all its neighbors will stop pulsing as well. And so there's a lot going on in these animals, but yeah, I do think they are interacting. They're obviously interacting. We are starting to look at these things as well.
Yes, there was a question in the back.
Yes?
Do you know the Reynolds number of the little barbs coming off of each tentacle? roughly?
So,
Well, I'll tell you why I'm thinking, and we can talk later,
Okay.
is we looked at copepods, which are crustaceans that have seedy with barbs on them.
Mm-hmm.
And the flow is very similar to what you saw. And when they bring the appendages apart, they go slowly like yours do, and the water doesn't flow through the gaps.
Right.
Because they're below a transition.
Right.
Then during the return stroke, they go faster and the water does flow through, so they can get net directional motion enhanced by that mechanism. So it'd be fun to talk about whether your guys might be using that too.
Right, and this is something Laura and I have actually discussed because we know this happens at that Reynolds regime. Yeah, so what we have done in these simulations, which I didn't mention, is modeled currently the bristles as a solid plate because we do think they're in the Reynolds regime where they're moving fluid back and forth. But this is something we are hoping to kind of do a separate study because we can't include, it would be too computationally expensive to include these in these simulations. So already these simulations are being run on 32 cores for about 5 days to do a 3D simulation. And so the thought of adding in the modeling and the bristles would be really expensive, but we'd like to do it independently.
Yeah, one more question. Maybe you can plug your,
Have you thought about looking at flows internal to the polyp?
So, like in,
Well, imagine that